I'm Steve Leavitt, and I study lichens, and lichens are these fascinating symbiotic systems where you have an algae and a fungus that interact together to form these miniature ecosystems. And my role is to look at these systems and try to identify the evolutionary relationships among these different symbiotic partners. So I work on the system of lichens called camouflage lichens, and these are incredibly common lichens. They're found on bark and sometimes on rocks. One of the greatest things about lichens is that almost any level that you look at them, they're fascinating. I mean, you can go out and see a cliff face that's splashed with all these colors of lichens growing on the cliff face, and it's beautiful. You can see a tree that's covered in lichens, and you can pull out your hand lens or your dissecting scope and take off a single specimen and start looking at it. And under a magnification, single individual is just as fascinating as the entire cliff face. Once we collect a specimen in the field, the next step is to bring it back here to the museum, to the lab, and we'll prepare the specimen for the molecular portion of our analyses. And we'll take the DNA, run it through a series of processes, and by comparing a wide array of DNA sequence data, we're able to actually distinguish and infer that there are multiple species hidden within. There's a bit of evangelical passion as I try to share the knowledge or the beauty of these lichens. And it's really fascinating to go out there and kind of take this look at a system or these organisms that are so predominant and so common, but they're so overlooked.